All right. Hello, everybody. Hi, Jane. Thank you so much for joining us. Welcome to Synthetic Biology, Biodesign, and Society with Dr. Jane Calvert. So my name is Allison Irvine. I am the Program Manager of the Biodesign Challenge.
And I'm Dan Grushkin. I'm the Director of the program and also Director of GenSpace. And this is Vina, who is joining us just recently this year. A lot of you already know her.
Hi, everyone. So as you all know, this is our first webinar in a series, and we just wanted to let you all know about a few more upcoming webinars before we started. So on February 14th, which is just a week from, February 27th, which is just a week from today, we have a webinar for the Animal-Free Wool Prize with PETA, Stella McCartney, and Straight Out Capital. And then on March 14th, next month, at 12 p.m. Eastern again, like today, we're doing Intro to Designing with Biology with Dr. Karen Hogan and Dr. Orkan Talhan. So we hope to see you all there. Make sure you mark your calendars for those as well.
Right, and if you can't make it, we're recording these sessions, so you can come back to our website in a couple of weeks and visit and see what we've said then.
Yes.
So I'm just going to give you a little introduction about how this session is going to go. So Jane will give a talk for roughly 45 minutes. During her talk, please feel free to use the Q&A window to ask questions. At the end of her talk, we'll refer to the Q&A window and moderate a brief Q&A with Jane, and hopefully she'll answer your questions there. So with that, I'm going to hand it over to Allison, who will introduce Jane. I'm very excited about this. Jane has been an influence on the Biodesign Challenge and really a lot of our thinking for so long. So this is really exciting for all of us.
Yes. So yes, to introduce Dr. Jane Calvert. She is a social scientist based in the Department of Science, Technology, and Innovation Studies at the University of Edinburgh. Her current research focuses on attempts to engineer living things in the emerging field of synthetic biology, which raises intriguing questions about design, evolution, and life. She is also interested in the governance of emerging technologies and in interdisciplinary collaborations of all sorts. She is the co-author of the interdisciplinary book, Synthetic Aesthetics, Investigating Synthetic Biology's Designs on Nature, published by the MIT Press in 2014. Welcome, Jane.
Thank you.
Thank you very much.
We're going to go off so we don't have to see our faces, and we'll pop up during the Q&A.
So you're going to disappear, are you?
Yeah. Yeah.
Okay. Well, I'll be talking to a blank screen. But hello, everyone. It's very nice to be here. A bit odd since I only have your names, but I'll just imagine what your faces look like. And thanks very much for the introduction. I'm based in Edinburgh, which looks like the picture on my slide. You should come and visit. And as Alison said, I'm a social scientist, and I work in a field called Science and Technology Studies. And since this is a kind of sort of odd field, small field, I think you might not have heard of it. So I thought I'd just give you a brief introduction or definition of science and technology studies. It's the study of the social, philosophical, historical, political, economic, and cultural dimensions of science and technology. So basically, I'm a social scientist and I study science. So science is the thing that I'm interested in. And in science and technology studies, in STS, we don't see science and society as these two separate spheres that kind of influence each other from afar. We see them as very interconnected. So everyone knows that science is part of society, that's obvious. But also we think that society is part of science. So the way that the science develops is heavily influenced by social factors. And I'll try and explain a little bit about what I mean by that. So I hope, can everyone hear me? I hope, just type if you can't or if anything's unclear. And type your questions, like I said, and I'll come back to them at the end. But kind of depicting science and society in that separate way is going to be wrong anyway. It's much more interconnected and mixed. And this is something that we're interested in STS, is looking at this kind of interconnection of science and society. So I am a social scientist whose interest is in the field of synthetic biology, which is an area I've been studying for the last 10 years. And the reason I find synthetic biology really interesting is because it attempts to apply engineering principles to living systems. And I find this kind of interaction of biology and engineering really fascinating because biology often resists this. So my current research project is called Engineering Life, and I'm investigating this kind of contentious intersection between biology and engineering. And as Alison mentioned, I was also involved in a project called Synthetic Aesthetics, which is why I came to have an interest in biodesign and bioart, and I'll talk about that a bit later. But first, I want to briefly introduce the field of synthetic biology because it's going to be the topic that I'm speaking to today. This is a poster that was used for the first ever synthetic biology conference in 2004. And as you can see, this idea that it's a kind of medley of biology and engineering is represented in this poster with the cogs turning into cells, or perhaps the cells turning into cogs. It's unclear which way round this interaction goes. And here's a poster from a conference that was several years later. And here again, you can see this beautiful representation of the idea that biology can be engineered. And actually, that poster is on the wall behind me, which you probably can't see. But here we have a much more kind of idea of precision engineering, something that's much more delicate and refined. So there are many definitions of synthetic biology around, but the one that I like and which is used quite a lot is the design and construction of new biological parts, devices and systems, and the redesign of existing natural biological systems for useful purposes. And as biodesign challenge people, I hope you'll notice the word design in this definition. So we have both the design and the redesign, which figures really prominently in this definition of synthetic biology. And if something is designed, then it becomes a very interesting topic for social science, because once something is designed, we can start asking a range of questions about it, like for what purpose is it designed well or not, and who is it designed for? So these are the types of questions that I'm interested in asking of synthetic biology. So that was a kind of introduction to three things, to STS, to synthetic biology, and to design. Very briefly, and now I'll just give you an outline of what I'm going to say for the rest of my time. So my talk is divided into two. The first is on the social dimensions of synthetic biology, and this is because I think it raises issues that are relevant to anyone who wants to design with biology. And this part of the talk will be based on a book chapter that I wrote with a colleague of mine called Emma Froh. Then the second part of the talk will be on the Synthetic Aesthetics project, and something I'll talk about then is what interesting things can happen at the intersection of different disciplines, specifically in my case, synthetic biology, social science, and art and design. Right, so I hope that all makes sense to everyone. I'll start now on the first part, which is on the social dimensions of synthetic biology. So like I said, feel free to type your questions and I'll come back to them at the end. So one of the things that strikes you when you study synthetic biology is that there are many reports that have been published on the field and its social and ethical dimensions. This is just a snapshot of some of the many reports that have been published over the last few years. And myself and my colleague Emma Froh, because we are very interested in synthetic biology, we read most of these reports and we found that a lot of them were quite repetitive and they repeated similar topics. So they talked about things like the ethical implications of creating life, about speculations, about future harms and benefits, about the public acceptance of this technology, and about concerns about risk and regulation. But we decided that this was actually not really getting us anywhere, that we were kind of going round and round in circles and that we were just thinking about the same topics again and again. So in this chapter, we tried to what we call reframe the discussion, so start to think about these things in slightly different ways. So we have four kind of reframings. The first is from social implications to social dimensions. The second is from speculation about risks and benefits to more grounded anticipation. The third is from public acceptance to the public good. And the fourth is from regulation to governance. So what I'm going to do is just briefly talk you through each of these four shifts in framing. And the reason we do want to reframe in this way is because we think this opens up a more productive space for thinking about the social dimensions of synthetic biology. So the first shift is from looking at the kind of ethical implications of the field to its social dimensions. And one of the reasons why we wanted to put forward this shift is because if you're thinking in terms of implications, you're thinking in terms of what happens after the research has been completed. Whereas if you think about dimensions, you think about what happens before, during, and after the scientific research. And just to give you an example of something that is more of a dimension than an implication is the use of analogies and metaphors in the field of synthetic biology. And I'll just give you an example of one of these, which is quite widely used in the field. And this is the idea of a chassis. And in synthetic biology, this metaphor, which is borrowed from mechanical engineering, is used to describe the context into which engineered biological parts can be put. So in other words, it's another name for the cell. So one of the questions that interests me as a social scientist is what happens when you start calling a cell a chassis? What does it enable and what does it suppress? Does it encourage us to ignore the unique features of living things? Or does it help us think about how we might better engineer with biology? And you can apply the same type of analysis to many different analogies and metaphors used in the field. Another one that's very common, which is represented by this quite beautiful picture, I think, is the idea of engineering in terms of Lego blocks, that we can kind of make living things out of standardized interchangeable parts. Again, we can ask questions of this Lego metaphor, what it implies, what it enables, and what it suppresses. But these metaphors are not just about language, they're also about power. So just to quote from the sociologist of science, Susan Lee Starr, she says, power is about whose metaphor brings worlds together. So metaphors can be very powerful. And I don't think it's an exaggeration to say that synthetic biology research is really influenced by the metaphors, and they are shaping the types of research questions that are asked and the type of applications that are likely to emerge. So I think it's really important that you think about the metaphors that are used and that are inspirational for your biodesign projects. The second shift is from speculation about risk and benefits to more grounded anticipation. And this was kind of based on a slight frustration that Emma and I had about how there was lots of talk about how synthetic biology was going to save the world or it was going to destroy the world. So there was very kind of hyped up discussion, but not so much kind of grounded thoughts about what actually might happen in the future. And both speculation and anticipation are about the future, of course. And the future is very important, and the future always has been important, even in the past. So this is a really interesting example of kind of thoughts about the future in the past. This is an article called Biology in an Age of Synthesis from 1971, which is way before synthetic biology as a field got off the ground in the early 1990s. But if you read this article, it really says things that are so similar to the things that synthetic biologists say today. For example, there's talk about making plants that will fix their own nitrogen, about designing microbes that will colonize other planets, and also about replacing the chemical industry with a more sustainable biological one. So these are all things that we find very much in the discourse of synthetic biology today, but people seem to be a little bit blind to history and not to realize that these things have been talked about for many, many years. But the kind of expectations about the future are very important in emerging fields like synthetic biology because they do require an incredibly visionary momentum in order to command investment and collaboration. So you have to kind of express your hopes about the future of the field in order to get people on board. So I'm not kind of criticizing these hopes and these expectations, but just noticing that they are kind of important. But to some extent, they're often, like I said, a little bit ungrounded, a little bit exaggerated. So there was lots of talk of synthetic biology being the next industrial revolution, and there still is, but it doesn't take much digging to find that there are lots of competitors for this title of the next industrial revolution. So, for example, nanotechnology is the next industrial revolution, 3D printing as well, the maker movement could be the next industrial revolution, the Internet of Things, cloud computing. I'm sure you can think of many other things that are the next industrial revolution. Also, there's some ambiguity about how many industrial revolutions we've had. The article I showed you before said synthetic biology was the third one, but some people say we've already had 5. So where does biology fit in this? And also, should we really be aiming for the next industrial revolution? Should we be aiming for things like, this is a picture of Manchester during the Industrial Revolution, the second one, according to the picture I showed you before, and you can see the factories spewing pollution into the air. So should synthetic biology really be following in the footsteps of these industrial revolutions? Or should it perhaps be trying to do something a bit different and something a bit more sustainable? So industrial revolutions have obviously resulted in great benefits, but also in tremendous equality and environmental destruction. So should synthetic biology be building a different and more sustainable future? This brings me on to my third reframing, which is about moving from public acceptance to thinking about the public good. And when you talk about many new technologies, people often shift to like discussion of public acceptance, and they often say that the reason people don't accept new technologies is basically because they're ignorant about them. So what we need to do is we need to teach people about science and technology, and if people understand it, then they will be positive. And this is called the deficit model, because it thinks that there is a deficit in people's understanding, and it conceptualizes the lay mind as an empty bucket into which the facts of science can and should be poured. The problem with the deficit model is that it has been shown to be incorrect. So decades of social scientist research have shown that attempts at increasing public knowledge have not led to increased support for science. And you can kind of think about this in terms of countries and social groups that are well educated are not necessarily more positive about science than those that are less well educated. Also, in respect to genetically modified organisms, or GMOs, which is something that synthetic biology is often compared to, there's evidence that more knowledge about GMOs makes people more skeptical or polarized, not less. So we can't predict the effects of trying to educate people. Also, the deficit model is usually accompanied by assumptions that what really worries people with new technologies is risk and safety. But actually, often that's not really the case. If you talk to people, for example, again, about genetically modified crops, they'll often say that the things that they're concerned about are things like the monopolization of technology by large corporations, the implications of industrial food distribution and trade, and threats to indigenous food culture and rural farmer livelihoods. But these types of things often never even enter the discussion if the focus is just on risk and safety. So it shows that we shouldn't necessarily assume what people are going to be concerned about. Also, we shouldn't assume that all new biotechnologies like synthetic biology will fall into the same patterns as genetically modified crops. And this is a point made by this article by Torgerson and Schmidt, who talk about the different ways in which synthetic biology can be framed. So one way is definitely the GM crops frame, and GM is often regarded as being an elephant in the room in discussions of synthetic biology. It's always in the background in some sense. But the thing about GM crops is that it often portrays technology as being a site of conflict. It's often connected with what's called the fear of the public. So this idea that the public are going to be scared and we should be scared of the fact that the public are going to be scared. And this is something that the social scientist Claire Maris has called symbiophobia phobia. And in this frame, the public is seen as an obstacle in the path of technological development. Another frame, though, is the nanotechnology frame, and this picture is one of my favourite nanotechnology images. It's from St Pancras Station in London, where apparently they have nanotechnology to clean the glass in the ceiling. The thing about nanotechnology is that in this frame, technology is perceived as being kind of progress, and it's been marked in the UK and other countries by lots of upstream public engagement, so lots of engagement with people at the early stages of the technology, and this has been widely regarded as a success. The next frame, which is another completely different one, is the information technology frame, and here technology is not something that's kind of done by people in big scientific institutions or industry. Technology, well, information technology obviously is done in those contexts, but technology can be seen as a form of personal empowerment. And in this frame, we get people thinking of technologies in terms of gadgets and personalized devices like phones, and ideas of playing, hacking, do-it-yourself biology are all connected with this frame. So here we have this idea that technology is something that can be democratized and something that many people can participate in and benefit from and contribute to. And so the interesting thing about this frame is it also challenges the idea that there's this kind of passive, homogeneous public who either accepts or rejects technologies. Instead, there's something much more active and involved going on with particular users. So the point here is that there are many different ways of thinking about new technologies and thinking about new developments, and the same with biodesign. We don't know how it's going to be interpreted and how it's going to be understood. But whichever way it's framed, there's a big emphasis on the importance of public dialogue around emerging technologies at the moment. And the idea is that this should not just be one-way communication, which is the kind of model of the deficit model, where you just tell people about the technology, but two-way dialogue with genuine deliberation and negotiation, where you can really think about what people, we can understand what people think about the technology and what their concerns are and what their excitement is. And one of the reasons for engaging in public dialogue is to try and further promote the public good. And this is something that this report on emerging technologies kind of tackled, this question of what is the public good. And there's no one simple answer to this question. Instead, the report argues that it requires a kind of ongoing negotiation and context-dependent discussion in the public sphere. So in order to know what the public good is, you have to engage with people who have interest in the technology, either as users or as people who might be affected by it in some way. And it's only through this kind of public discussion that you can work out what would be good. So there's no one group which can kind of say what the public good is aside from everyone else. And this takes me to my final shift of this first half of the presentation, which is from regulation to governance. And the reason that we thought this shift was important is because a lot of the reactions to new technologies are that we need to kind of regulate the risks, and that's how we should deal with them. But we think it's more interesting to think about broader forms of governance. And this is because when technologies are still emerging, it's very difficult to calculate their risks in advance or to predict their unintended consequences. So this is Waddington's epigenetic landscape, which is kind of used for thinking about the development in genetics. But the point I want to make here is that we don't know which direction the ball is going to roll down. So in emerging technologies, we just don't know how they're going to turn out. So how can we regulate the risks when we don't know what these are? So arguments are made that in these situations, we have to just allow for uncertainty instead. And we have to, because we don't know what's going to happen, we have to encourage flexibility and diversity in technological developments. And this can avoid lock-in to a limited range of paths, which might turn out to be unsatisfactory in the future. And a form of governance which is rising in prominence across Europe and in the UK and in some other countries is responsible research and innovation. And this synthetic biology consortium came up with a definition of responsible research and innovation, which is the incorporation of ethical, societal, and environmental considerations into synthetic biology design. And again, there's the word design here, which I think is significant, that ethical, social, and environmental considerations can be integrated into biological design. And the thing about responsible research and innovation is it starts again from this point that because we don't know what the risks are, we shouldn't just think about the risks and regulating them. We should instead shift our thinking to thinking about the motivations and purposes that drive the research. So the idea here is that responsibility isn't retrospective. It isn't about trying to think about who's to blame for things that have happened in the past, but it's prospective again. Instead, it's about being responsible towards the future. And for me, the kind of quotation that summarizes this idea of responsible research and innovation is, what kind of future do we want science and innovation to bring into the world? And I think this is quite a kind of inspiring way of thinking about how to do scientific research. And if we want to bring a particular future into the world, then we have to think about alternatives. We have to kind of open our imaginations to different possibilities. And this is where I'm going to make the connection between the first part of my talk and the second part of my talk, because I think critical design is a really interesting way of thinking about responsible research and innovation, because both of these are interested in new ways of thinking about the world. And as I'm sure many of you know, critical design is a branch of design that explores possible technological futures through objects that provoke debate. So that will lead me on to the second part of my presentation, so I finished the first part, and it would be great if someone could just type something, just so I know that you're there. Just to say that I've covered a lot of stuff in this presentation. Great, you're there. And I know I've rushed through it very quickly, and there's lots in there. And if you would like to find out more, there is this book chapter which has much more detail and some more examples, and the Biodesign team, oh good, lots of more people are saying they're here, excellent. The Biodesign team will make this chapter available to you if you're interested. And also, if you'd like a longer version of this talk, my colleague Emma Farrow, and we wrote the chapter together, she's actually got a nice, I think it's like an hour-long version of that particular chapter that you can go and watch. Okay, thank you for people for saying you're here. And I've got a question coming up, which is great, which we'll come back to you later. Okay, so moving on to the second part, which is synthetic aesthetics. So like I said, I ended up being involved in this project, which resulted in this book that was published in 2014. But the project itself started much earlier. It was in 2009. There was a call for an event, a sandpit called New Directions in Synthetic Biology. So I applied for this, and to my surprise, I got into it. And so what we did is we were kind of put together for a week in a conference center just outside Washington, DC. This very hothouse environment. We had to come up with proposals. At the end of the week, $10 million was awarded towards the projects, which was divided up amongst several projects. We didn't get it all ourselves. And so what the whole point of this sandpit was to be inventive and to kind of develop ideas that wouldn't normally come out through peer review. And so what resulted from this sandpit was this synthetic aesthetics project, and this involves myself, another social scientist called Pablo Schifter, and then two synthetic biologists, Drew Endy and Alistair Elphick, and then Daisy Ginsberg, who is a critical designer. And I just want to explain a bit more about Daisy's involvement in the project. So I first met Daisy in 2009, or yes, it was 2009, I think, in the iGEM competition, which is the International Genetically Engineered Machines competition, which some of you might have heard of, which is a synthetic biology competition. So basically, the rumor of the iGEM competition was that someone, a couple, a man called James King and Daisy Ginsberg were walking around with a suitcase, and everyone was like, have you seen the suitcase? Have you seen the suitcase? What's in the suitcase? And so if you were lucky enough to find them, they would open up the suitcase to you and they would reveal this, which as you can see is coloured poo, and it's fake, it's not real, I should say. It was made in a fake food factory. But the reason they developed this is because they'd been working with an iGEM team who was interested in making E. coli express colours that are visible to the naked eye. And so their idea was that you would drink a probiotic yogurty drink, that it would interact with your gut bacteria, and that it would produce an output that was easily visible in the normal way. And so this was a really interesting exploration of the interaction between the body and synthetic biology, and it produced something which is much more kind of visceral and slightly disgusting, and which is very different from the ways we normally think about new technologies with kind of shiny pipettes and lab benches and things like that, and test tubes. Also, this project was a speculative design project, so it wasn't real. It was an exploration of what could be done, but many people thought it was real, and I think this is not the fault of the designers. I think this is the propensity of some people to want to believe certain types of technological futures. And so this is the Daily Mail, which is a terrible paper that's published in Britain, but it says that that scientists create yogurt that changes the colour of your poop to diagnose illness. So basically, the Daily Mail thought this was a real project when actually it was a speculative one. So I think this is an interesting issue for the Biodesign Challenge about the boundaries between the fantastical and the real. So basically, we, like Daisy's work, so we invited her to be part of the project, and she was very influential in the way that the synthetic aesthetic project developed. And so what we actually did is the project is we advertised for participants. We got lots of people who were interested, and we ended up with choosing just 6 artisan designers and 6 synthetic biologists, and we paired them, and they did exchanges. So they spent 2 weeks in each other's workspaces. And it's quite normal for artisan designers to spend time in the laboratory, but it's less normal for scientists and engineers to spend time in the art studio. But we were very keen that the exchanges should be reciprocal and that it shouldn't be one way, but it should be 2 way. So we forced the scientists and engineers to spend an equal amount of time in the art studio. And these are the 6 projects that were part of synthetic aesthetics. We tried to get people from a broad range of different areas of art, design, and synthetic biology. But because I don't have much time today, I'm just going to briefly talk you through 4 of the projects. So the first of these 4 was a project between an architect called David Benjamin, who's at Columbia University in New York, and a synthetic biologist, Fernando Federici, who's at the time was at Cambridge in the UK. And they were very interested in issues of scale and how you bridge them between the very small scales of biology and the big scales of architecture. But what they weren't interested in was things like this. They weren't interested in just taking biological shapes and making them big. And the way they expressed this is that they were interested in the logic of biology rather than the form. And by the logic of biology, they meant the way that biology computes and solves problems. So something that really inspired them in this was this experiment where this yellow dot is slime mold, and then the white dots are food sources. So basically you let the slime mold grow and it connects the different food sources in a very optimal way. But the way in which the dots were arranged was to replicate railway stations in the Tokyo railway system. So the way the slime mold grew drew a very kind of efficient map of what the Tokyo railway system perhaps should look like if it had been grown according to the logic of biology. So this was the type of thing that really inspired them in their work. And another thing they did is they took 3D images of cells. These are xylem cells from the plant science lab. And they put these 3D images in architectural software, and then they subjected them to evolutionary algorithms in architectural software, which resulted in new forms, which they then 3D printed. So this is a really nice kind of interaction between the different scales of biology and architecture and how these things could talk to one another. The next pair is a speculative designer from Amsterdam and a protocell scientist who is based in Italy. And the protocell scientist was very interested in making very simple living things out of non-living constituents. So not that he's succeeded in doing this, but he's very interested in ideas of what it is for something to be alive. And the thing they decided to explore in their joint project was the question of whether inanimate machines are only an interlude in history. So what they noticed is that animate machines like horses, like oxes, have been really central to human history for many centuries. And even when inanimate machines took over, like steam engines and automobiles, these are actually very much modeled on animate machines. For example, horsepower is the standard of measurement we still use for vehicles. But they asked, well, what happens if we go back to animate machines, which is something that synthetic biology promises, but it promises something at a much more microbial level. So we're working with cells that actually grow and mutate and evolve and change very rapidly. So if we are going to be designing animate machines in the future, we're going to have to be designing with Darwin, designing with the forces of evolution. And in their project, they even explored the idea of an evolving steam engine. So I think for me, what's really interesting about this project is that it raises a question which is at the heart of synthetic biology, which is to be alive, a living machine must evolve. But if it evolves, it may no longer possess the stable, predictable properties that we expect of our machines. You wouldn't want your microwave evolving on you, for example. The next pair I'm going to talk about is Hideo Iwasaki, who is a scientist, a cyanobacteriologist from Waseda University in Tokyo, and Oren Katz, who's a bioartist from the University of Western Australia. And they wanted to use cyanobacteria in their collaborative work. And cyanobacteria is the green stuff in this tray. And the thing about cyanobacteria is they do lots of great things. One of the things they do is they break down discarded computer chips, or they could be used to do this. And this might be a big problem with lots of discarded computers all over the place. And what they noticed is that one of the products of this breakdown would be gold, but another product would be fool's gold. And fool's gold looks like gold, but it's a useless substance. It doesn't have the characteristics that we value of gold. So what they thought they could do is engineer cyanobacteria to preferentially produce fool's gold. So they wanted to engineer it to produce something useless. And they wanted to do this because they wanted to draw attention to the fact that synthetic biology is so oriented around applications and utility. So what they wanted to do is engineer futility into cyanobacteria. But the main focus of their project was to explore the kind of relationship between geological and biological time. And again, this is inspired by the cyanobacteria, the organism in the project, because cyanobacteria operate at many different time scales. So, for example, they have very fast metabolic cycles, like all organisms, but they also have circadian rhythms. They also operate on day-night cycles, which are slower. But then at a much, much slower time scale, there were also the organisms that were responsible for converting the Earth's atmosphere to oxygen. So they are responsible for life as we know it, basically. And the early Earth was probably covered in these thrombolites, which are actually rocks made from cyanobacteria depositing minerals, and they can be found in some places in the world now. This is where they visited as part of their residence, which is the saline lakes of Western Australia. So the reason they were interested in geological time is because it puts all human activity, including synthetic biology, into a totally different perspective, because we'll all just end up as another layer in the rock. So they wanted to use time as an instrument of humility to challenge the very kind of present-oriented focus of today's biotechnologies. And the final project I'm going to talk about is one that was between the smell artist Cecil Tolas and the synthetic biologist Christina Agapakis. Christina was at Harvard at the time, and Cecil Tolas is based in Berlin, working kind of alongside international flavors and fragrances. And Cecil's a smell artist, and this is her laboratory, her smell lab, which is an amazing place, and she does all sorts of things with smells that I don't have time to go into, but it's very interesting. So in their joint project, they became very interested in our contradictory relationship with bacteria because, as you know, bacteria are on us and in us and all around us, but yet we seem to regard them as dirty and hazardous, and we seem to try and get rid of them with antibacterial cleansers and things like that. So the means through which they decided to explore this relationship was cheese. And this is because they discovered that many of the bacteria that are found on cheeses are also very similar, sometimes identical to those that are found on human skin. And this is probably due to artisanal cheese-making practices where people kind of put their hands in the cheese when they were making it. So as part of their project, and apologies in advance, this does make some people feel a bit sick, is that they extracted bacteria from different parts of their bodies and other people's bodies, and they used it to make cheese in the laboratory. And they made these different cheeses. So there's armpit cheese, toast cheese, hand cheese, and nose cheese. And we couldn't eat the cheeses because they hadn't been washed, which is a kind of technical procedure to remove harmful bacteria. But we could smell them. And it was really interesting, the different smells. So the armpit cheese was yogurty and fragrant and very nice. The toe cheese was disgusting and really powerful and pungent. And the other cheeses had different bouquets. So this project, I think, raises really lots of interesting questions about our relationship to bacteria, about the human microbiome, and also about the boundaries between our bodies and our food, because bacteria don't distinguish between them in the same way that we do. But something else that their project did that I really found interesting is that they kind of concluded that rather than modeling synthetic biology on computer engineering, cheese making might be an engineering paradigm that allows for the design, construction, and maintenance of complex living worlds performing incredible feats of metabolism. So they are suggesting a new metaphor for synthetic biology, which is cheese making. And another thing that they kind of drew attention to, because they actually sequenced the bacteria that lived on the cheeses, and there are of course many, many different bacteria, because cheeses are a kind of vast collection of different bacteria and yeast and other things. And they noticed that in nature, living things are always part of dynamic, interactive, symbiotic communities. And in fact, one of the things that really struck me on reflecting on the synthetic aesthetics projects is they all drew attention to features of biological things that make them different from normal engineered things. So Sasha and Sharif drew attention to evolution. Oren and Hideo thought about the temporality and the timescales of living things. And Cecil and Christina kind of talked about symbiosis and interaction of different organisms. And what I think all the residents did, including the ones that I haven't had time to talk about, is they thought about designing with biology rather than design of biology. So designing in a way that is sympathetic to the capacities of the biological substrate, perhaps. So perhaps this is something that biodesign can do. And I think they also drew attention to a kind of humility of working with living things through their work. So just going to end on some further reflections on the Synthetic Aesthetics project. For me, one of the most exciting things about this project is its interdisciplinarity and the fact that it brought together synthetic biology, social science, and art and design in a way that was very non-hierarchical, so nobody was the boss in a sense. And I'd never worked with artists and designers before. This is my first exposure to art and design, and I found that they really had a lot in common with me as a social scientist. So we were similarly interested in opening up synthetic biology to a broader range of perspectives. We wanted to enable alternative technological pathways to be explored, not just thinking in normal ways. And we wanted to also question the dominant futures that might be imagined by powerful groups. So this, I hope you can see, kind of relates to this idea of responsible research and innovation that I talked about earlier, about thinking about alternative ways in which things could be developed. And I kind of drew this conclusion through interviews with many of the people involved in the project. And this is just one interview quotation from Oren Katz, the bioartist. He says, "I think there's an urgent need for artists to explore major issues that are being raised by synthetic biology, because if we're not there at the very beginning, it'll be too late to be able to show alternative ways in which this knowledge can be applied." So it's this kind of emphasis on showing alternatives that I think is really interesting here. And of course, this project wasn't just about social science and art and design, it was also about synthetic biology too. And I think together, the three different disciplines, we really came up with something at this intersection that none of us could have done alone. And I've called this an emergent form of critique in an article that I've written recently. And by critique here, I don't mean something that's kind of negative and kind of damning. I'm actually using an idea from the anthropologist and philosopher Bruno Latour, who says that the critic is not the one who debunks, but the one who assembles. The critic is not the one who lifts the rugs from under the feet of the naive believers, but the one who offers the participants arenas in which to gather. So I suppose this is what we wanted to do in synthetic aesthetics. We wanted to offer people arenas in which to gather, places in which to do things that they wouldn't have done otherwise. And so what I hope the project produced was new things, new ways of doing things, and new forms of interdisciplinary collaboration. And I think that is my hope for the biodesign projects as well, that they will produce these kind of new ways of thinking. So that is at last everything I had to say. Sorry, apologies again, lots and lots of stuff here. These are the references that I used in both of the part 1 and part 2, and this is just thanking my funders and my current research project that pays my salary at the moment. So I'll put it back on the references in case people are interested in that, and I would be happy to have to answer any questions now. So maybe do Dan and Alison want to come back online? Hi, yeah.
Yes, we're back.
Thank you so much,
Jane. We were here the whole time. I did have a quick question for you. Given that all of the students who are participating in the Biodesign Challenge are really at the beginning of their exploration of working with, working to understand biology, right, and how it might be a design medium or something you can design with, as you said. Is there advice you can give them, having been through this process, that can help smooth their journey as they go from this to a project in June?
Oh, what do you mean by smooth their journey?
Meaning, like, how should they be thinking about biology, or are there certain things that they should be paying special attention to as they pick their design topic, as they explore their brief, if you will? Yeah,
I mean, like I said about synthetic aesthetics, I think the distinctive features of biology are really interesting. So some synthetic biologists seem to want to make biology into cogs and wheels and standardized parts, but actually biology does a lot more interesting things than that. So it is context dependent, it does evolve, it does change. Biological organisms exist with other organisms. So all these features, I think, can actually be inspirational. And I think this is what I learned from the artists and designers on the project, is that they really engaged with biology as a new material which was not like other materials. So does that help?
I hope so. Yeah. We'll find out in June, I guess. Yes, I think it does. I mean, there's this whole question of whether or not these properties of living things is a feature or a flaw, right, when you're talking to a designer. Obviously, there's no point in working with a biological system if you see the biology of the system as a flaw. Before I move on to the questions, I just had another question. Have you seen an evolution in the way that synthetic biologists have been thinking about the biology and the biological systems?
Yeah, I think so. I think there's been much more recognition of the difficulty of engineering biology and of having to kind of work with biology instead of like working on it. I think using evolution in synthetic biology has become much more accepted, and so in a way synthetic biology has perhaps become more like traditional biological disciplines. So, which is interesting, but one of the things that drew me to synthetic biology in the first place was this real aspiration to engineer with biology. So I wouldn't want that to be lost, because that's something that kind of keeps me interested in the field, I suppose.
I'm gonna ask some of the questions that we're seeing popping up in the Q&A. So there are two that are very much about public engagement. So I'm gonna ask them both. So FIFA asks about science communication. So, how do you, or where would science communication be most effective from the point of view of public good, which I think is a really, really interesting question. And then the other one was more about, again, about public engagement with the projects. And the question was, how do you engage with the public, and what advice would you give on how to discuss slash frame such types of projects? So again, also, how do we shift the frame to a public good frame? And what does that actually look like in practice?
Yeah, I mean, this is the thing about the public good is not something you can set out and you can say, I know what the public good is and I'm just going to go and pursue it. You have to engage with people who are, I think, relevant to your particular project. So I think this will often arise out of the project. So if it has an application in a particular area, I think talking to people who are going to be the users of that or who might not like it or something would enable you to kind of think about how the public good is best served. So FIFA's question about where would science communication be most effective? Within that is another question about what is effective science communication? So like I said, with the deficit model, the idea is that effective science communication is getting everyone to love the technology. But for me, that's not effective science communication. I think it would be more about learning from people about what they value or what they don't like and trying to integrate that into the design. So this is something that might be a useful thing for teams to do, is to start even at the early stages thinking about who might be the users, who might be regulating this technology, and perhaps try and engage with them in their work. And I know you have a prize for fieldwork, so that seems to fit with that. But I do think that just instead of thinking about public acceptance, thinking about a public good is just a really handy way of kind of changing the questions you want to ask. So rather, thinking like, how can this technology serve the public good? Even though there's no easy answers, it kind of reorients the whole discussion in a quite a valuable way, I think.
Yes. Yeah, thanks for the shout out for the prize. So yeah, very much the prizes was really written to encourage students to, number one, figure out who they're serving, and then really get to know them before or as they're developing their projects, so that the project is really about those people.
Yeah. In social science, we kind of say that there is not a public, there are publics. So there are lots of different groups. So it really helps to kind of break them down and not think about what would the public think, but think about who are the relevant publics, who are the relevant groups. And they might actually be other experts. So you can engage very productively with experts in, for example, environmental science or medicine, who have different kind of views on the world.
Great point. Yeah, it's very true.
Yeah, so something Minje is asking is about, I think the metaphor she's asking about is the comparison of biology to machines. Now, exactly, the chassis. And also, I mean, even when we talk about DNA, we talk about it as code, which I think is always in some ways a reference to kind of human-made language or human-made code or computing code often. I think that the question is really, how to have, well, I'm going to split that question into two. Have we mined that metaphor enough in the history of biotechnology, or is there still room to mine? And is there a better operating way to think about it that scientists and obviously artists?
Yeah, so the metaphor I talked about at the beginning is a chassis, which is the thing that provides the kind of framework for a car. And that's one of my favorite metaphors in synthetic biology. I think it's so rich and so interesting because it's a real challenge to think of a cell as a chassis. I've heard some like microbiologists with expertise in a particular organism like really cringe when they say, oh, Bacillus is a chassis, and they go, no, it's not a chassis, it's a cell, So it kind of maybe tries to reduce that cell to a mechanical thing. But we should also think about the fact that the cell itself is a metaphor. So I think it was, I'm going to get it wrong. Was it Hooke or someone who looked down a microscope and saw cells and thought those are just like the things that monks sit in, so that's where he used the word cell. So you can never escape from metaphor, but I think you just have to be sensitive to the implications of your metaphors. And I think this is where artists and designers can be so useful, because they can develop new metaphors, and that would be a wonderful thing, I think, to come out of the Biodesign Challenge. If people had new ways which were not just kind of copying the metaphors that we already have in other fields, but actually bringing new metaphors in. Of course, they're going to come from somewhere. They're not going to come from nowhere. Yeah, going beyond the kind of ideas of engineering or of code, and thinking of biology differently, perhaps.
I think that leads into another question that I had, which is, when we built the Biodesign Challenge, I mean, it was largely based on the ideas that we were reading about in synthetic aesthetics. So whether or not, you're happy with that evolution of the thinking or not, I leave to another conversation. But I wonder, if you could, we're bringing artists and designers to this subject, really now all over the world. We're in 9 countries right now.
9 countries.
I wonder if you could kind of outline some of your aspirations for them, meaning like how would you want them to push what you've started, and Daisy and the whole team push what you've started forward? How would you like to?
Yeah, big question. So it wasn't me who started it. Like I said, I'd never worked with artists and designers before. I kind of accidentally ended up in synthetic aesthetics, but it was a wonderful experience, and partially because it was so interdisciplinary. And this is what I was saying, that you don't have to do this alone. If you come together with people from different disciplines and different backgrounds, you might get something kind of really interesting coming out of that. But I suppose for me, an aspiration of of future biodesign is that it shouldn't lose its critical strand. And this is why I talked about an emergent form of critique that emerged from the Synthetic Aesthetics Project. And what we together, and this included the synthetic biologists as well, so it wasn't just a criticism of synthetic biology, we all were kind of getting quite frustrated with the way that we saw the field shutting down, becoming narrow, becoming kind of industrialized, becoming like biotechnology as usual. And we wanted it to be something different and something special and something more inventive and imaginative. So we came up with a shared metaphor of a wedge in the door, that we kind of used to describe the project, and then we'd used to describe the book. So we were like, well, even if people don't read it, they can use it to wedge a door open. So it kind of became something that we shared across the project team. And I think that like this idea of of being able to think differently and having that freedom for thinking differently, I think that for me is a kind of aspiration. And also teams come up with such amazing imaginative ideas that you would never expect. So yeah, that I think people have it within them to kind of challenge established ideas through bioart and biodesign.
I love that.
Great. So I think that those were our questions, unless we have one more. They're starting to come in. Now they're starting to roll in. This one is
A two-part question.
a two-part question. So how do you think critical and speculative design can further facilitate the public engagement with science from a futures-making perspective? And then the second question is, there seems to be a second wave of synthetic biology startups moving towards application fields such as cosmetics, food, etc., rather than health and energy. Besides PES, hold on, moving down. Do you think that design contribute to more divergent thinking in early stages of scientific research? So yeah, I think this is a beautiful question, which is, how early can we bring design into the scientific process? Does it reach before the applications that go out
Yeah, I
into the world,
think
and how
definitely
do we
early
make that
stages. I
possible?
mean, this is why in synthetic aesthetics we involve people in the laboratories. So it was really at these very early stages, and partially that's why they came up with such crazy projects like the cheese, I suppose. Also, synthetic biology understands itself as a design discipline. So I suppose we see that design, a potential for that design thinking to enter in very early on. This is Irem's question? I'm probably pronouncing it wrongly, sorry. But I actually can't see the whole question. I probably just got it. I can only see the first bit of the question. But but I think you managed to read it out to me, Dan, didn't you?
So yeah, you have to click on the question. So I'm realizing now that we have a setting where the audience can't see the questions.
Right.
So sorry about that technical
Difficulty.
difficulty. However, I'll just go back to that first part of the question, which was about public engagement with science, moving science from a futures-making perspective. Or let me just read it. How do you think critical and speculative design can further facilitate the public engagement
Yeah, I
with
think
science from a futures?
I think again, it depends who the public is, but I think in terms of making futures and making diverse futures, which is something that critical and speculative design does really well, I think this is where other people, so maybe the public or certain members of the public, could actually contribute to thinking about things in different ways and kind of bringing in a broader range of imaginations to bear, perhaps. Yeah, so I hope that addresses that.
Well, thank you, Jane.
Thank you so
This
much.
was absolutely wonderful. And I think all of us are really, really grateful that you've chosen to join us and take your afternoon
Yes, thank you so much for your
with
time.
us.
Yeah.
I'm going to just remind the audience that next Tuesday, February 27th, same time, same channel, we'll be talking to representatives from Stella McCartney, PETA, and Stray Dog Capital about animal-free wool. So that should be interesting.
Yeah, so can I just say thank you to the
I'm really looking
participants?
forward to having
It's
you all
been
back for that one.
like it's a
Thank
shame
you
not to see your
again,
faces,
Jane.
but I really appreciate your comments and thank you. It was very nice to kind of meet you in a kind of just seeing your names way.
Wonderful. Yes, thank you so much.
Well, we'll see you all soon,
Yes.
hopefully.